This is a production of Cornell University.
So I'm going to start out and remind you about what I talked about last year, which was also the practical haplotype graph. And so last year I talked a lot about building the practical haplotype graph in sorghum and how we were trying to use it in a breeding program which is run by Gaël Pressois and based in Port-au-Prince, Haiti. Okay, and so we've used the practical haplotype graph in that program for genomic selection a couple times now. And in the last year, really one of the things that we've done is we've tested the practical haplotype graph with different genotyping methods. And so today, actually, what I wanted to talk about is how we've tested the PHG and share some of those genotyping methods and the results with and without the practical haplotype graph. Okay? So to start out, we have our nice genomic selection scheme on the right here. And in any GS scheme, we have this point where we need to genotype the progeny population. And we have a lot of options for genotyping. And so this list on the left is absolutely not an exhaustive list, but it does list some of the possibilities that we have. But really what we want is a technology that's going to be cheap, it's going to be fast, and we also really want a technology that's fairly easy to use both across breeding programs and also across species. And so when we start to subset down this list based on those criteria, we can actually cross out a lot of the things that are on this list to begin with. But two of the ones that are left are Rampseek or Rampseek and Skim Sequencing. And so these are two genotyping methods that I have used with the practical haplotype graph. And so that's what I'm going to be presenting for you guys today. Now, last year, I put up a slide and I said, okay, the practical haplotype graph is really designed to be an imputation tool. And so it's been designed to work with a lot of different types of sequence data. And I used GBS as an example of that. I put up GBS data and I showed, okay, we can use GBS for genomic selection in Gaël's breeding program. And we can also use that same sequence data with the practical haplotype graph in Gaël's breeding program. So this year I'm not going to show you GBS data with the practical haplotype graph, but I am going to use that GBS data as our baseline. We know that GBS is a really an effective technology, and so we're always trying to get prediction accuracies with new technologies that are at least equivalent to that, okay? So I'll start actually by talking about Skim Sequence. And when I talk about Skim Sequence, what I'm really talking about is a protocol that was designed by Nextera. And essentially they take a TN5 transposase that jumps into the genome at different positions randomly across the entire genome. And then we sequence out from those positions. And so that gives us a very sparse initial data set. And it's also completely random. So we're not capturing one specific or consistent marker set with this technology. But it is really cheap. It costs less than $10 per sample. And so it is a viable option or something that we'd like to be able to use with a breeding program. And so the question at the bottom then becomes, does imputation with the practical haplotype graph make it possible to use this technology? So I'm going to put up this slide first. This is a plot very similar to one I showed you last year. And both in this plot and in plots for the rest of my talk, if the box is blue, it's GBS data. So that's just the baseline, okay? GBS works well for prediction accuracy in Gaël's program. Now, if I add in boxes for that Nextera Skim sequence data, that's those green boxes on the bottom. And you can see that, yes, this technology is less than $10 per sample, but it's not giving us anything. It's completely useless for prediction, okay? And that really comes down to the fact that because this is based on random sequencing, there's no consistency, there's no overlap in positions that we're actually sequencing. And so this is where the practical haplotype graph can come in. If we take that sequence data and we run it through the PHG to impute missing information, we actually get these dark green boxes here. And so you can see that the practical haplotype graph is actually helping us to restore that prediction accuracy, and we can get an equivalent prediction accuracy, approximately the same number of markers as we would have with GBS, and we can do it at half the cost. And so the answer to the question then is yes, with Skim sequencing, we can use the practical haplotype graph for effective genomic selection. So the second technology is Rampseek, or Rampseek. This is an amplicon-based sequencing method, and it targets specific regions, so it does give you fewer markers, but it's offered as a service by a couple different companies, and that means it's very little work for breeders to use, and so we like it for that reason. And so Rampseek, when we put up a plot like this, you see that using Rampseek positions or amplicons on their own actually works really well. We're already about the same level as GBS with this light purple, those light purple boxes. And I do want to point out that this is targeting about 2,000 loci across the genome, and they've been designed specifically for sorghum, but not specifically for Gael's program, okay? Now, when we take that sequence data and we run it through the practical haplotype graph, we get these dark purple boxes. And that doesn't look so good because you can see we're not improving our prediction accuracy, we're actually really decreasing them. And so that tells us two things. The first is that in Gael's program, the 2,000 markers that we're capturing with Rampseek is enough to do effective genomic selection. And the second thing is it's telling us that with this technology right now, the practical haplotype graph is probably just adding random noise. And so this is something that actually the programmers have changed actually how we're imputing these genotypes. And so in the next couple of months, I'll be looking at if those changes have actually improved this kind of plot. But the answer right now is that no, with Rampseek, the practical haplotype graph doesn't really help us and isn't really necessary at all. So that brings us to this nice point where we have a couple different options for genotyping. And the option that a breeder chooses really should be designed or should be chosen based on both their breeding goals and the budget that they're willing to spend. So I'm going to build this plot a little bit up on the right here. On the x-axis, we have the number of SNPs. It only goes to 3,000, but you can imagine we could extend that fairly significantly. And then on the y-axis, we have cost per sample. Okay? So I'll be plotting data that comes from Agriplex Genomics. That's one of the companies that produces this Rampseek amplicon-based sequencing. And as I plot those, I've also colored, hopefully you can see it okay, the background of this plot to show you at that location in the graph, what is our best sequencing option cost-wise, okay? And so just as an example, if we had a point where this X is on the screen, that means we want more than 2,000 markers and we're willing to pay more than $15 per sample. And so GBS is a good genotyping strategy in that particular case. Does that make sense? Great. So let's add some data. If we start adding data, we can see that if we're working in a species that has a lot of breeding efforts going on, maybe we expect that across all breeding programs there could be 80,000 or even 100,000 individuals that are being genotyped. Then we're following the blue and purple lines on this plot, and actually Rampseek is going to be our best bet. We can get about 2,000 markers from Rampseek, and we're still under that $10 per sample goal that we'd like to be hitting. If we're working in a species that has more moderate breeding efforts, maybe we have 10,000 or 25,000 samples that we expect to be genotyped, then we're following the orange and green lines on this plot. And we can really decide between Rampseek and Skim sequencing with the practical haplotype graph. Both will give us a cost-effective option for genotyping, and we really want to be paying attention then to how many markers we need, because the cost for 1,000 markers is fairly equivalent. And then last, if we're working in a small breeding program, we're following the red line here, and Skim sequence with the practical haplotype graph is really going to be the most effective option for us in this case. I also want to point out that the data from Agriplex only goes up to 2,000 markers, but I sort of tried to extrapolate out from that, and that's what the dotted lines on this plot are. And so if you extrapolate out, if you need more than 2,000 markers, either for your breeding program or because you want to be doing other genomic analyses, then something like GBS or Skim sequencing with the practical haplotype graph is going to be a really good option for you. So that gets us back to our original question. When is the practical haplotype graph most useful? And the answer is 1, if we're working in a species that has fairly small breeding efforts, the PHG can help get us cost-effective genotyping. Another is that if we're working in medium or large breeding programs, but we want more than 1,000 or 2,000 markers, the PHG can also help fill in some of that marker information that we might want. And then the third is, if we're working in a breeding program or set of programs and we want to have a consistent set of SNPs, the practical haplotype graph can actually output a consistent SNP set. And so that can help with that kind of consistency as well if we need it. So I am about out of time. Hopefully I've convinced you that the PHG does have its uses, and we have used it effectively in Gael's breeding program, although I don't have time to talk about that here. There's a lot of people that have worked on this, and so I do want to acknowledge everybody in the Buckler Lab, everybody in Gael's program at Shibas, and then Jeff Morris and Kabede Muleta at the Morris Lab, who have also put a lot of time into this project. But I'm going to wrap up. So if you guys have questions, I'd be happy to try and answer those now, or we can talk afterward as well. Thanks.
So were those plots, those bar plots, are those from the same taxa, or is that like the next generation selection?
Good question. So the question was, all of the box plots I showed, are the taxa being shown here the same? And the answer is yes. This is actually all data from the training population that Gael made, and so there's about 250 taxa in that data set, and it's consistent across all of the marker technologies. Great. Thanks, guys.
This has been a production of Cornell University, on the web at cornell.edu.